Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm a part of the team here at SYNGAP Research Fund. Our presentation today discusses oligos that target translation to restore SYNGAP1 levels. I have the pleasure to introduce today's speaker, Dr. Brian Dickinson from the University of Chicago. Brian earned his BS in biochemistry from the University of Maryland and his PhD in chemistry from the University of California at Berkeley. And after a Jane Coffin Childs Memorial Postdoctoral Fellowship at Harvard University, he joined the faculty at the University of Chicago in the Department of Chemistry in the summer of 2014. In 2019, he was promoted to associate professor, and in 2023, he was promoted to professor. The Dickinson Group employs synthetic organic chemistry, molecular evolution, and protein design to develop molecular technologies to study and control chemistry in living systems. The group's current primary research interests include: 1, developing new evolution technologies to reprogram and control biomolecular interactions; 2, engineering RNA targeting biotechnologies as new therapeutic platforms; and 3, leveraging chemical biology to study biomolecular interactions. The motivating principle of the Dickinson Group is that chemists' ability to create functional molecules through both rational and evolutionary approaches can lead to new breakthroughs in biology and biotechnology. A recorded version of this webinar will be available on the SRF website under the webinars on the family menu. By the end of the presentation, you will have the opportunity to get your answers to your questions. We'd love to hear from you, so please write your questions in the Q&A below. And for those of you just joining us, welcome. And again, our speaker is Brian Dickinson. It's now my pleasure to turn things over to Brian. Thank you.
Great. Thank you for having me today. It's really nice to get to chat with a really diverse audience about some of our work. Let me share my screen now. So this might be a little bit different than the way these have normally run, in the sense that our lab is really a platform technology lab. And I'm going to talk today about some of the kind of platforms we've been developing to target gene expression in a variety of contexts, but I'm going to try to highlight how this relates to SYNGAP1 in particular. So I'm going to start with a rationale of how we think about SYNGAP1 and why we're interested in going after it as a therapeutic target. And then a lot of the talk is going to be just about technologies we're developing. And then we'll come back at the end to show you how we're developing these specifically for SYNGAP1. Okay, so yeah, my lab is at the University of Chicago in the Department of Chemistry, a really diverse team of researchers thinking about designing molecules that study and control chemistry in living systems in a variety of different contexts. So starting off with the rationale within SYNGAP1, as this audience knows very well, there's a really complex set of pathology that emerges from deficiency of the SYNGAP1 protein. A lot of human suffering emerges from what is in many ways a kind of innocuous, quite boring protein. So SYNGAP1 protein is a RAS GPase activating protein. Deficiency in this protein results in this kind of complex set of pathological phenotypes like intellectual disability, developmental disorder, severe autism, et cetera, as well as an epileptic phenotype. And as you all know, there's currently no treatment that directly targets the underlying cause of the disease. So from a kind of human biology perspective and medical perspective, this is really complicated and quite tragic. From a chemistry perspective, what we can recognize is it's a really frustratingly simple problem. This is some of our data with a collaborator here in Chicago that looked at the SYNGAP1 protein levels from a healthy matched control patient versus a patient who's heterozygous deficient in the SYNGAP1 protein, and it's just 50% too little of the protein is present. So from a molecular standpoint, we know the problem. There's just too little of this protein being made, roughly 50% too little. But that results in really complicated human pathology because of that deficiency in that one protein product. So this gets at the idea of haploinsufficiency diseases. If you look at, humans have 20 or 30,000 different proteins, and each of those proteins need to be expressed at the right time and place within the body in order to drive physiology. And probably for a lot of proteins, there's a relatively narrow band of kind of healthy physiological protein expression levels, and too much or too little of that protein can result in disease. In the case of deficiencies, where one of the two copies of the gene is mutated, either with a premature stop codon or some sort of deactivating mutation, one way we can view the kind of molecular basis of disease is that there's just 50% roughly too little of that protein being made. And we're kind of outside of that kind of band of the kind of healthy expression levels from the kind of normal patient population that you would see. And this is called a haploinsufficiency. So ideally, if we could have a technology that could restore those protein expression levels back into that physiological band of expression, that would be a way to directly go after these sorts of diseases. What makes this really complicated, though, is this kind of healthy expression level could be highly cell type dependent. Maybe one cell needs one level, a different cell needs a different level. In the context of neurological diseases, maybe neurons need the protein expressed, and maybe even at different levels, depending on the subtype of neuron, but other types of cells maybe shouldn't have it expressed. So it's really difficult to kind of get this Goldilocks scenario where you need the protein present, but not too much or too little, and have it in the right place at the right time. That's a very challenging problem. So getting at what my lab does, we study gene expression regulation in a variety of different contexts. We have evolution technologies where we're using evolution to design complex molecules that interact with biological systems. We do chemical biology of protein lipidation. We target RNA with engineered biological systems, and we map RNA localization. That's the kind of four main project areas, all geared at this idea of thinking about how gene expression is regulated at the RNA and biomolecular level. So for today, thinking about how we can target haploinsufficiency diseases and diseases of gene sufficiency, I'm going to talk about our subgroup on controlling protein RNA interactions, and in particular, designing bifunctional molecules that target gene expression at the RNA level, and how we think that this could be one strategy to think about developing targeted therapeutics for deficiency disorders such as SYNGAP1. So if you think about aberrant gene expression, I already said that in some cases, the problem is too little of a particular gene is being expressed because one of the copies is mutated. DNA to RNA to protein. Protein composition is, in most cases, the underlying cause of human disease. And therefore, it's unsurprising that almost every single drug on the market targets proteins, in most cases by inhibiting them, whether that's antibody therapeutics that sequester targets or small molecules that bind to proteins. We have a very protein-centric view of drug discovery. Of course, in cases where the problem is not an overactive protein, but actually something that's missing, small molecules and biologics that target proteins can't really help in most cases, right? Because if the problem is a lack of something, not too much of something, then this kind of inhibitory mindset isn't really viable. We live in a brave new world where one can think about now going in and making specific changes to the underlying DNA of a cell to correct genetic basis of disease at the DNA level. And that is really exciting. But I think in some cases, this is going to be really problematic to do in some tissues and in some contexts. So our lab is really interested in targeting RNA therapeutically for a couple different reasons. RNA is the intermediate in the kind of gene expression central dogma. The RNA content of a cell is cell type dependent. So if we target RNA regulation at the transcriptome RNA level, then the effects of that target will be reflected by the content of the RNA within the cell. So in other words, if we're making a change to a cell based off of an RNA that's expressed in the cell, the level of that RNA will kind of dictate the response that that cell will have. So a cell that has a lot of the RNA will have more of a response. A cell that doesn't have the RNA will have less of a response. So it kind of lets the cell guide its own response to the therapeutic based off of its RNA content. And we think that's really one way to get around this challenge of getting cell type dependent expression, which is needed in a lot of these sorts of complex human diseases. So it's highly regulated, it's cell type specific, and also RNA regulation is very tunable. We can kind of fine tune gene expression up or down at the RNA level if we can harness these regulatory processes. So a lot of classic biologists viewed RNA as just this intermediate. It's really not. A lot of gene expression is highly regulated at the RNA level. So just to give you a sense of what that means, that the amount of protein made per RNA is a very regulated process, and that's something that we're going to talk about targeting today. The half-life of an RNA and how long it lives within the cell is also a regulated process, and therapeutic strategies like RNAi target the degradation process of RNAs to retune the half-life of an RNA. One way that cells regulate RNAs is by chemically modifying them with a variety of chemical marks, and these chemical marks then serve as adapters for other proteins to bind to the RNA and regulate it. RNAs are spliced. In some cases, splicing can drive disease. In some cases, splicing can be targeted to restore homeostatic protein levels. RNAs are also edited, and the bases can be changed to alter the proteogenic nature of a particular RNA. And then all these processes, as well as localization, are guided by protein-RNA interactions, which control, say, export from the nucleus and trafficking, et cetera. And collectively, all this regulation is often referred to as epitranscriptomics, which basically just means all the different ways the cell tunes gene expression at the RNA level. And if you look at this from an engineering perspective, we can see all this regulation is coordinated by really two main processes: protein-RNA interactions, so effector proteins binding to RNAs and altering their regulation, and then cellular localization, where the RNA is present and how it's kind of guided through the cell. And we're studying both of these processes, and really, again, from an engineering perspective, we view all this complex regulation as possible ways to control RNA regulation if we can design systems that specifically harness each of them. So we started on this a while ago, where we looked at one of the regulatory networks within the cell that targets RNA, and that's the M6A pathway. So this chemical modification here, called M6A, is present on the A base of RNA, methylation here, and cells have dedicated methyltransferases and demethylases, which dynamically mark subsets of the mRNAs within a cell with this chemical mark. That chemical mark then allows those RNAs to be recognized by these so-called reader proteins, which bind to those M6A modified transcripts and alter their regulation, either their half-life or their rate of translation or other features of that RNA. And what we realized a few years ago is that the reader proteins that the cell naturally uses to regulate RNA are kind of bifunctional molecules. They have an M6A binding piece, and then an effector piece that kind of recruits other factors to alter the regulation of the RNA. So we had this idea of, what if we could remove the M6A binding domain from this protein and make it into something that we can engineer, so that the protein doesn't bind to all the transcripts with this M6A mark, but instead binds to the specific transcript that we want to regulate. So the way we did this is we engineered a Cas protein, this Cas13 system, which is an RNA-dependent RNA binding protein. It's the RNA cousin of Cas9, which you're probably quite familiar with. And this guide-dependent RNA binding protein basically just binds to a transcript that we want based off of the programmable nature of this guide RNA. And what we showed is that we can then pluck these effector domains out of their native context, engineer them onto these Cas13 systems, and turn them from M6A dependent readers into guide dependent readers, so that now we can drive these responses to specific transcripts within the cell. So for example, we can get guide dependent gene activation. That's analogous to what we would want to do in the context of something like a haploinsufficiency such as SYNGAP1, or we can get guide dependent destabilization of the transcript and degradation in a programmable way. So basically, this just showed this idea of taking these natural RNA regulatory proteins and directing them to transcripts that we want using these engineered guide-dependent systems. So these CRISPR systems have a lot of advantages. Of course, they're being deployed therapeutically, but there are a couple challenges with thinking about how we would deploy them therapeutically. One is they're quite large, which precludes delivery with like an AAV-based therapeutic. And they're also potentially immunogenic, and that's going to be potentially problematic in some applications in human therapeutics. And many of us already have antibodies against Cas proteins in our systems because we've been infected with microbes that have them as part of their proteome. So we asked the question a few years ago, can we rebuild a programmable RNA effector protein complex that's as small as possible and critically not built out of these kind of microbial protein systems, with the idea that that would potentially circumvent some of these immunogenicity issues that we were quite concerned with. So this was our original design based off of the Cas13 system. We have the Cas13 protein that's deactivated with a guide that binds a target of interest. We then engineer it with these various effector proteins that then are driven by that guide to target an RNA and then elicit some sort of an effect on that RNA. And what we realized is that this kind of protein engineering concept is relatively simple. And we thought that perhaps we could build a system that does all of this, but that uses only components derived from the human proteome. So kind of an all human protein-based system. The idea was, what if we took a small high affinity RNA protein complex from the human proteome and simply displayed a guide RNA off of that? Now, this wouldn't be very happy in a cell because it will look like a viral RNA and will be recognized by an innate immune response. But the way Cas systems deal with this is through non-specific protein RNA interactions. So we have specific protein RNA interactions that drive interaction with the guide, and then non-specific interactions that kind of shield that guide sequence from interaction with other cell components. So we thought we could just engineer that onto our system with a small non-specific RNA binding protein. So now we'd get this guide-dependent RNA binding protein complex. We could then put all of our effector domains we want to interact with onto that system. And if it works, ideally now have a guide-dependent RNA binding protein that elicits an effects based off of that guide sequence. And we call this technology the CRISPR-Cas inspired RNA targeting system, or CIRTS. And what's really powerful about this is, rather than using microbially derived proteins and repurposing them as therapeutics, we can just take all of the kind of human proteins present in our body and use those as kind of a collection of starting materials to then think about how to engineer them together to build these guide-dependent RNA binding protein systems. So while the fusions between them are not natural, at least all the kind of core building blocks of the CIRT system are built out of human proteins, but really repurposed for this new role. So we can design these systems. So here's an example of a simple one. This is a nuclease. So it's a guide-dependent RNA nuclease. We can show that it binds an RNA based off of its guide, and it can degrade an RNA based off of that guide. So it's a guide-dependent RNA nuclease, very analogous to the natural CRISPR systems, CRISPR-Cas systems, but now in this case, built out of entirely human protein components. We can then append all sorts of different vectors onto it. So we can make it into a nuclease. And in these data, I'm showing you in human cells, an off-target guide in gray versus an on-target guide in black, showing different effects. So if the effector is a nuclease, we can get guide-dependent RNA nuclease activity and degrade the target RNA. Very similar. This is our system, the CIRT system. This is the natural Cas system, very similar activity. We can put a translational activator. This again would be the kind of sort of effect we would look for in a SYNGAP1 guide-dependent activation. Now we can put a destabilizer that deadenylates the RNA. We now get a guide-dependent destabilization, or we can put a base editor to cause single base edits on the RNA. And now we have a guide-dependent RNA editor. So we can kind of, again, hijack all these different types of regulation that occurs at the RNA level and now guide that regulation to specific transcripts to alter gene expression at that RNA level. We've more recently shown that these can work in vivo in a mouse model and with small molecule control. So this is really a more synthetic biology application, but what we can do is we can split the RNA binding protein component of the CIRT system from the effector protein, so that in the kind of a basal state, nothing happens to the RNA. But then when we add a small molecule, in this case, this plant hormone, abscisic acid, that induces the dimerization and the kind of full assembly of the system, which then causes regulation to occur at the RNA level. And in this experiment, we have a liver editing assay where there's a luciferase from fireflies expressed in the liver of these mice with a stop codon in it. And what we can do is, in the case where those mice are just growing, they don't have any luminescence emerging from their livers. But when we add in this small molecule abscisic acid, we can turn on our RNA regulatory system and cause single base edits on that transcript, which restores this firefly luciferase gene. And then we see light emerging from the livers of these mice. And what this is really showing is that we can even engineer kind of exogenous control over this RNA regulation, thinking of what about disease cases where we want to kind of fine tune gene expression based off of the kind of symptomatic development of a patient, we can now add small molecule exogenous control over those RNA regulatory processes. And this also gives us the ability to kind of test these systems in kind of a mouse model. So now what we're doing, this is ongoing work in the lab, is we're trying to take these SERTs constructs and further expand their activity, both activators and destabilizers in cases where we need this kind of tight band of homeostatic gene regulation. And SYNGAP is one of the targets that we're currently screening different types of effectors for, and specifically for activators in this context. But we have kind of a range of human diseases we're looking at where we think this sort of approach could be valuable. And we're right now going through mining the human proteome to find all sorts of different effectors that can work well for destabilization or activation, and really trying to understand, how do we design those systems? How do we deploy them? What sort of matching occurs between different types of effectors with different types of targets, and really trying to expand that technology. I should also note, for full disclosure, I did start a company that's trying to develop this sort of technology for therapeutic applications. So in a summary of this first part, these are protein-based systems that can target RNA regulation, and they really fit into a broad array of different exciting technologies that target RNA regulation. I think since the COVID, we started this before the COVID era, but I think now post-COVID, the world is a lot more comfortable with thinking about genetic therapies and RNA targeting technologies. So I think we're really excited about seeing how far we can push these to these diseases where there just isn't a simpler alternative, where we really need to have this high level of control. I think RNA is a really good application of that. So like I said, we're improving the performance of these systems, expanding them, developing preclinical models in a couple different of applications, and working on delivery of these systems, primarily through AAV-based delivery strategies. So I want to switch gears now and talk about a completely different project that really emerged from some of our thinking about RNA regulation with these CRISPR systems, but moving toward a different modality. And as a chemist, I really think about the molecules we're using to build technologies out of. And so far, we've been talking about these protein RNA complexes, which is one strategy, primarily used with like a viral-based delivery system for human therapeutics, like an AAV-based system. That's how we're thinking about those. But we were interested in moving toward other modalities, and I'm going to talk next about oligo modalities. And then we're also thinking about even moving beyond that towards small molecule modalities. So thinking about where, what sorts of chemical material makes sense for the particular application in terms of disease and what we actually want to do within a cell. So I'm going to switch away from these protein-based systems and talk about something totally different, which is an oligo-based system, but still trying to target RNA regulation. Now, if we think about oligos, meaning RNAs or RNA-like molecules, oligos have now been engineered to target almost every aspect of RNA regulation. So we can target the degradation of an RNA with RNAi or ASOs. We could target splicing with ASOs. That's a really interesting therapeutic technology right now, and that's being deployed for SYNGAP1 in some applications as a strategy. We can control the chemical modification state of RNAs, even with RNAs, and then we can even direct base editors to RNAs with oligos, and these are so-called ADAR recruiting oligos, which is another really exciting area in biotech right now of therapeutic technology development. So the one area, which is what we would ideally want to do for diseases like SYNGAP1 that we really can't effectively do with oligos at the moment, is target translation. Basically to tell the cell to make more protein from the RNA that's already present within the cell. Now, there is, and the reason this can work is the amount of protein made per RNA is a highly regulated process. I already showed you that with some of the CRISPR-like systems I described about how we can activate translation from a target RNA. Now, there is one technology that's currently being commercialized. It's been developed over the last decade or so that's really interesting. This is called SYNAPs. These are long non-coding RNAs that bind at the stop codon of a target RNA. And activate translation through still somewhat completely not worked out mechanisms. But what's really neat about these SYN elements is you can direct them to the start codon of an mRNA and boost protein production from that RNA. So like I said, these are being, there's a small startup developing these therapeutically. And I think that's really interesting. they're still quite long, so they tend to be in the hundreds of nucleotides long. So not a true oligo. Probably AAV would be the best strategy for something like this, but kind of an alternative to the protein-based systems I talked about. So we set out a goal for ourselves to develop an oligo-based system that's small enough to be chemically synthesized, that programmably targets RNA regulation from in a mechanistically defined way, so that we know the effects, we know how to design it, and we know how to make it work. Okay, so if we think about how one would target translational regulation, if we think about translation, we have an mRNA that's expressed in the cell. We then have what are called initiation factors that bind the RNA and then tell the cell to assemble ribosomes on it that make that RNA and translate it into a protein. So our simple idea was, if we know that these initiation factors are kind of rate limiting in this process, what if we design a bifunctional RNA, which we call a translational activating RNA, or tRNA, that has a guide-dependent binding modality on it, some sort of linker, and then some sort of an initiation factor recruitment domain, such that if we can bind initiation factors onto that RNA, perhaps we can tell the cell to put more ribosomes per time on that RNA, making more protein product. Now, in particular, we want to design these systems that bind to the 3' UTR. That's on this side of the RNA. And the reason for that is what we learned from some of our work with the CRISPR-like systems is that the 5' UTRs for many genes are both small and crowded. And we really wanted to design systems that could work in the 3' UTR of an RNA. Now, the reason you can get translational regulation in the 3' UTR, which we already knew based off some of the natural regulatory mechanisms that occur for translation, is that the RNA kind of exists in this loop structure. So binding things over here can, at least in 3-dimensional space, at least in our mind's eye, be sort of near the translational start site. So that can have an impact on translation even when bound over here. But it's helpful because the 3' UTR has a lot of free real estate to interact with, a lot of unpaired bases with low degrees of secondary structure that we can get oligos to bind to reasonably effectively. Okay, so now the key here is, where are we going to find these RNA elements that bind initiation factors? And of course, nature has evolved these for us. So viral RNAs, like hepatitis C virus and many viral RNAs, use what are called irises, internal ribosome entry sites. These are structured RNAs that, once the RNA is inside a cell, hijacks the initiation factors that are endogenously expressed in that cell to turn that viral RNA into a viral proteome. These are also present in some human RNAs for alternative start sites. Now, these viral RNA elements bind different initiation factors and promote translation. In nature, these are always cis-acting elements, meaning that these RNA structures are part of the RNA that is going to be translated into protein. And that makes sense because you can see in these schematics, we line up all these proteins on the RNA, and then translation starts at the start codon. So what sort of, and in the tRNA strategy, we're going to be asking to have them function in trans, meaning we're going to be delivering them to another RNA and trying to promote the translation of an RNA that they're not even a part of. So what sort of indication did we think that that was even possible? So it turns out, if you look at some plant viruses, unrelated to these, some plant viruses have RNA structures in the 3' UTR. So still part of the RNA, but very far away in primary sequence space. But there are these 3' UTR elements called site elements that actually recruit initiation factors to promote the translation of that RNA. So at least in 3-dimensional space, very far apart, but still able to promote translation. And this gets at this central idea of how we think about biology, that's really all about local concentration and proximity. So potentially our hypothesis was, we can find the right types of elements to get them to just line up on the RNA in the right way. Perhaps that would be able to promote translation in trans in the tRNA design. And it turns out, after some screening, that was true. So we started with some simple luciferase reporter assays in human cells, where we had these tRNA designs where we just took full irises from a bunch of different viruses, engineered guides onto them to see if we could promote the translation of a luciferase reporter gene. So in gray is an off-target guide and blue is an on-target guide. So this class 4 designed tRNA with the class 4 iris, which directly promotes ribosomes, doesn't have an impact really on the translation of that mRNA. But you can see these class 3 and class 2, which promote all sorts of different interactions, promote the translation of this reporter. Critically, while we see increases in protein, there's no change at the RNA level. So these are translationally activating systems. So we then picked one of the more well-studied hits from our initial screen, which is from the hepatitis C virus, which is this RNA shown here. And we started doing studies to see what are the key interactions that drive this activity, what are the key targets of it, and can we shrink the system down to a more oligo-sized system. So through truncation studies, we found out that just this element up here, the so-called 3ABC domain, was enough to recapitulate much of the gene activation of the tRNA system. So we could take this 500 or so nucleotide system, shrink it down to just 120 nucleotides, which includes a 40 nucleotide guide, this linker, and this 3ABC domain shown here, and that alone could recapitulate much of the gene activation we were seeing. Based off of studies from other researchers, we knew a particular mutation, U228C, that in the natural virus iris element, that ablated one of its key interactions of this domain, which is binding to EIF3. When we make that mutation in the tRNA, which blocks EIF3 binding biochemically, we can see a diminishment in activity. So now we know one of the key initiation factor targets of the system that's responsible for part of the activity. And then we could take that kind of truncation understanding of what is the key feature of this particular iris and transplant that into other irises from other viruses. This CSFV and this PTV truncation also work. So we have kind of multiple structures from multiple viruses that we can kind of shrink down to a piece about this size here and have it function. So this PTV3AB element, which is actually the sequence I have shown here, that's the one that seemed to work most consistently. And that was kind of our lead tRNA design that we used to kind of move forward toward other studies. So we can then, that was all with a plasmid expressing the RNA. So now we want to actually deliver RNAs to cells to see if the system works in an RNA delivery assay. So now, to stabilize the RNA and keep it from being degraded, we put these kind of stabilizing hairpins on each end of the RNA. So we can then make the RNA in a test tube and then put it into cells to see if it works. So with this whole system here, we have the stabilizing hairpin, a guide RNA that directs it to a particular mRNA within the cell, and then this effector domain that binds EIF3 and promotes the translation of that target with a stabilizing hairpin on the opposite side. So in this experiment, we in vitro transcribe that RNA and then deliver it to cells. And in this case, we're targeting a tumor suppressor PTEN just as kind of a model target. And what you can see here by Western blot and then quantified is that we can boost PTEN expression with this RNA system. So we're now, again, using an RNA to promote the production of a particular mRNA into protein within a human cell. We can then do this in vivo, so we can package these into lipid nanoparticles, very similar to those that are used for the COVID vaccine. And we can inject these into mice. The lipid nanoparticles will tend to accumulate in the liver. And then we can ask, can we also promote the translation of this PTEN gene in the liver of mice? So we inject, wait 12 hours, isolate the liver. And what you can see again by a Western blot and quantification is we can promote the translation within the liver of a mouse as well. Now, with these kind of biochemically stabilized RNAs, the effect is relatively short-lived because eventually these RNAs will be degraded and the effect will be lost. So this is really a way to transiently activate gene expression. If one wanted a longer-term response, you'd want to install chemical modifications into the system, such as like is used with an RNAi type therapeutic in order to kind of stabilize that response. That's something that we're trying to work on in the lab right now. Okay, so next we wanted to go after a clinical model. And let's kind of finally get back to SYNGAP1 here. So with a collaborator, Al George at Northwestern, we were able to check the activity of these tRNA systems in iPSC-derived neurons from a haploinsufficient patient donor of the SYNGAP1 haploinsufficiency. So this is the first two blots here are the original data I showed you at the very beginning of the talk of a healthy matched iPSC-derived neuron versus a SYNGAP1 patient. And again, I told you that there's about 50% too little of that protein being made. So then we can grow these neurons in culture, package tRNAs in lipid nanoparticles now that deliver that technology to the SYNGAP1 mRNA, and ask if we can promote upregulation of the SYNGAP1 gene from the healthy copy of that SYNGAP1 mRNA to try to restore levels to the healthy matched control donor. So get about twofold increase in gene expression is the goal of this experiment. So we take those iPSC-derived neurons, add our lipid nanoparticles, encapsulating our tRNA into those, wait 8 to 12 hours, and then measure the SYNGAP1 levels with a Western blot. And we have off-target controls here with on-target tRNAs that activate the SYNGAP1 gene. And what you can see is we promote the translation. And what was really gratifying about these initial experiments is we actually promoted it to a level that was pretty near what we were looking for. So again, traditional gene therapy, when you're just overexpressing something, you don't have a lot of control over how much the gene is expressed. So you get levels that reflect how much of the technology is delivered to a specific cell. In this case, because we're just subtly upregulating gene expression, we don't dramatically boost protein levels, but we do kind of pump them up a little bit. And I think that that is really useful for something like these haploinsufficiencies, such as SYNGAP1. So then with that kind of initial kind of positive hit for SYNGAP1, we decided to really go after it and try to optimize the system biochemically to target SYNGAP1. So SYNGAP1 mRNA has a pretty long 3' UTR. And this is actually another really nice example of why we think targeting the 3' UTR is a much more widely useful strategy. If you look at the SYNGAP1 mRNA, it has a pretty small 5' UTR, about 202 nucleotides. And from our experience, there's not a lot of real estate to interact with there. There's a lot of bound proteins in that 5' UTR and not a lot of accessible sites to interact with. But we have, 17 or 1800 nucleotides in the 3' UTR with lots of different sites predicted to be unstructured. So the first thing we did is we took our initial guide, which bound right after the stop codon, guide 1, which worked reasonably well, and we just looked at other guide landing sites across the 3' UTR. And we found in particular, this guide 4, which binds about 200 nucleotides after the stop codon, seemed to perform the most reproducibly and to the highest efficacy in the system. So we then used this guide 4 to kind of further optimize the system around. So we wanted to shrink the system even further to kind of make it a more kind of compact system that's more aligned with the kind of what can be chemically synthesized in the lab. So our original system looked like this. Through kind of iterative rounds of truncation optimization, we were able to further shrink it down through a couple of different changes. First off, we didn't need 40 nucleotides to bind to a target of interest. We could truncate that down to 30 nucleotides without losing much efficacy. We also realized this linker was dispensable in this case, and some of this kind of stem here was also dispensable based off of truncation studies. So through all those extra kind of precision changes to the system, we're able to shrink this down so that the core structure is now only 95 nucleotides. So we're now below 100 nucleotides total of the core system. That's if it were chemically synthesized. In this case, we still have these kind of biochemically stabilized elements on the ends to kind of allow us to deliver it as RNA and look for efficacy in our biological models. So here is comparing our mini tRNA system to our original shrunken system. So now with those changes, we didn't lose any efficacy, but we shrunk the system down. This is looking at SYNGAP1 levels in 2 different contexts in N2A cells. This is a mouse cell line, or in HEK cells, this is a human cancer cell line. And in both cases, we can see a nice promotion of the SYNGAP1 expression. So as the final piece of data I'll show you, this is kind of the best experiment we've run so far. This is going back now to those patient-derived neurons to ask if we can restore SYNGAP1 levels with now this kind of what we call mini tRNA system, this shrunken system, again, packaged in lipid nanoparticles and delivered to those neurons. And again, we do all these experiments with a healthy matched donor. So that's this experiment shown here, this ++ genotype. This is the healthy level of SYNGAP1, the haploinsufficient patient-derived neurons, we have 50% too little. This is our mini tRNA with an off-target guide. It has no effect on SYNGAP1 levels, but when we have the guide delivering the system to the SYNGAP1 mRNA, we promote the translation. And again, when we look at the quantification, it really nicely lines up with the roughly twofold we're looking for in the healthy matched control. It's kind of a downstream marker of whether we actually altered the physiology of the cell by doing this. We can look at the phosphorylation levels of ERK. It's known that when you are deficient in SYNGAP1, ERK becomes hyperphosphorylated, and we can see that in these two bands here. When we restore SYNGAP1 levels with our tRNA, ERK phosphorylation goes down, and that can be shown here too. So we can not only see, this is a molecular phenotype, so not like physiology, but we can see at least the cell is responding to this upregulation of SYNGAP1 in levels that we would predict based off of what is known in the literature. So we're really excited about this result because it really looks like we're, at least at the molecular level, in line with what the needs of these cells are based off of this haploinsufficiency. So what are we doing with this now? So we're trying to improve the performance of this system, both in terms of reproducibility and size, and just really expand the number of targets we have to interact with. So we're testing other irises, working on engineering them, and asking about whether there are other targets that we can interact with. We have at least one other initiation factor that seems to be able to promote the translation of some target transcripts. So we're asking, when is one better than the other? In what context do they work? Do they have synergy or not? Those sorts of kind of fundamental questions. We're trying to push forward in preclinical models and targets, SYNGAP1 being one of the ones that we're most interested in this case. And we have some collaborators that are helping us kind of establish some of those preclinical models. And of course, for clinical application, critical for any sort of biotechnology is thinking about delivery. So whether it's an AAV strategy in the context of our CRISPR-like systems and our SERTs technology, or lipid nanoparticles or oligo-based technologies, in this case, we need to think about chemical optimization and other sorts of preclinical optimization to make these actually deployable as therapeutics. So there's a lot of work that needs to be done to move to that stage. But I think as we develop the technology and think about the sorts of needs of patients and where they are and what sorts of delivery strategies we can deploy based off the needs of those patients and the technology we have, I think that's a way to really move forward toward developing novel sorts of therapeutic strategies for, again, these diseases that really seem intractable. But I think once you have this kind of engineering mindset, you can start to at least envision how this sort of strategy can be used to target gene expression in a really defined and programmable and targeted way. So I'll end there. I want to acknowledge my team. I run a really eclectic mix of grad students and postdocs and undergrads that do all this research. I want to call out Yang Xiao, who is here, who's now a postdoc at Berkeley, who developed the tRNA technology, and also Riley, who's a current grad student in the lab who's developing our SERTs platform, including working on SYNGAP1 in the lab with that system. So stay tuned for some updates on that work. I also want to call out our collaborators, Al George at Northwestern, who is working with us on the patient-derived neuronal systems. Bill Green at UChicago, who's a neurobiologist who helps us think about the neurobiology aspects of it. Jimmy Holder from Baylor, who actually isolated these SYNGAP1 iPSC-derived cells, and then Sarush at UIC, who helped us kind of mechanistically interrogate the tRNA platform to some degree. So with that, I'd love to answer questions and tell you more about what we're doing, and I'd be happy to talk more in the future as well. So reach out if you have any questions or ideas. And thanks for providing me with this opportunity to tell you about our work today.
Okay, hang on. Thank you very much. I'm got an audio issue. Are you guys hearing my echo, or am I just
A little echo.
I know what's going on. Okay, that should have fixed it. Sorry about that. Wow, I'm speechless, which rarely happens. That was I love hearing about it when someone's been working on SYNGAP1 and we weren't paying for it and we didn't even know it was happening, and you've got such great results. So before I jump in with like a lot of questions, and then I saw your message, JR, that you can't talk on this one, but please type furiously if you can, if you've got questions. So first of all, I want to say, we do have a number of iPSCs at a CRO we work with called RareBase. I think we have 6 lines with same-sex family controls that if you want to expand your library, those are freely available. Like there's some transfer costs, but we can fix that. So if you're double clicking on SYNGAP and you want to expand the number of lines you're playing with, just know that those are available. Number one. Number two, I'm sure you're aware that Rick Hugonier's lab put out 2 mice with patient-derived mutations, and then we've actually made a 3rd one at JAX, which we're about to fund the characterization of. So there's 3 mouse lines that have a mutation in them as opposed to being knockouts, which actually gets to one of my questions about the 3' end. So when you've been putting, I think you've put this in mice already, but I'm not sure.
Not yet,
Like
no.
Okay, so when you put this in mice, I mean, I'm always curious, like in the old days, by which I mean like a few years ago, we just had knockout mice, right? And that was the model for the disease. But now as we're getting to these increasingly sophisticated therapies, like what you're talking about, it's a fair question. Like, okay, well, what is the knockout the right model? Like what's happening within the mutant allele, right? And would it make more sense to understand what you're doing? So the 3' end for the uninitiated who might be listening is for mortals who don't have PhDs is sort of the end of the protein, right? And what we tell parents,
End of the RNA.
The RNA, pardon me, the end of the RNA. What we tell the parents is like, look, when you have a stop codon, party's over. But with the RNA, since the 3, like what's happening in the protein truncating mutants?
Yeah.
And that's a leading question because you know what my next question is going to be, right? Which was what's happening in the missense mutants.
Yeah.
But so I just want to advertise that we have the cell lines and we have the mice in case you want cooler models to play with or more models to play with. But is it fair to think that your therapy is primarily focused on protein truncating variants?
It's a great question. So, yeah, and first for the iPSC, that's great. I'll definitely, so we're working really closely with Al George, who's really very connected, I think. So we'll definitely.
Well, I'm really glad to hear that Al George has gotten out of the channelopathies because that was just so boring. I'm glad to hear he's finally.
I don't think he's. Well, I think everyone. I mean, so I'm a technologist. So one of the reasons we were attracted to SYNGAP1 is, there's a lot of haploinsufficiencies, especially within the brain. There are all sorts of rare and semi-rare genetic disorders. And for all of them, it's always an open question. How much of the pathology is driven from the deficiency of the healthy allele versus the pathological allele doing something that it shouldn't be doing, kind of this so-called dominant negative effect. And for most diseases, it's not known. There's some combination probably of both. The mutant protein is doing stuff it shouldn't be doing, and the healthy protein there isn't enough of it, and the combination of those 2 effects leads to pathology. One of the reasons we got really excited about going after SYNGAP1 is it seems like SYNGAP1, more so than a lot of other diseases, is primarily driven, at least from my read, from the haploinsufficiency. Now, for a particular patient's mutation, we don't know, right? There could be a dominant negative effect also that is driving some of the phenotype that patient is experiencing. And then I think the question in terms of mouse model is, does the mouse model even recapitulate that? We don't know, right? But right now, when we target 3' ETR and we target translation, we're going to be it's not mutant specific, right? We're going to be targeting both the healthy copy and the unhealthy copy. So if by targeting the unhealthy copy, we just upregulate the bad stuff that's happening, that we can't avoid that right now. But we are going to be promoting the healthy copy. So this will be most effective for patients who are suffering from a true haploinsufficiency. And then, a question is, can we actually identify those patients? And do we know that that's the case?
Absolutely. I mean, you probably don't follow my podcast, but whoever's doing SYNGAP in your lab should be. And I just did an episode on missense mutants. And what I try to bang into patients' heads is all protein truncating variants are assumed to behave the same, right? Classic haploinsufficiency. But if you have a missense mutation, we should probably make a cell line, because when we start talking about therapies like yours, like that question will be genotype specific. Well, does it work in this one, and doesn't work in this one, and doesn't work in this one? And we're
absolutely
our misset. And I've had this conversation with people in government, and I've said I've been blown away when I hear about studies and research and clinical trials and people aren't genotyping, and they're like, yeah, we're with you. So I think this is a question that's going to keep coming up.
Absolutely. Just one other note about that that's actually important for when you make mouse models. I mean, for us, because we're targeting the sequence of the RNA, it also may be important to have the human knock-in mouse model, because the sequences we're targeting, we try to be strategic and pick sequences that are basically the same in the mouse and the human. But some of the things that can work really well in one, it's just the sequence is totally different and we're too divergent. So even the 3' UTRs can be very divergent between different species. So that's just another complexity in the system.
Good news, that's been made.
Yeah.
So we have a humanized mouse, and it's at the Prosser's lab made it at UPenn, and he's sent it to the JAX, and they're crossing it. So we're going to have a humanized mouse and a humanized head.
Yeah, that's great.
So yeah, I look forward to playing with that. You acknowledged that you had a company in the course of your conversation. To a comment and a question there. The question is, can you tell us what it's called, or is it in stealth? And the comment is, I think it's, you said that like it was almost like you were confessing something. And I think the opposite is true. Like, I think people as brilliant as you who have established labs and are doing science that are closely connected to or have your own companies are actually probably who organizations like ours should be preferring, because it means that you actually get that this has to be translated into something that can be commercialized, right? So I think there are people out there who are like, there's all this cool science and one day somebody else should commercialize it. And that's where I'm like, I'm done. Because I don't care. I love science, but what I care about is making therapies for kids. And so the fact that you are thinking about it from bench to, marketing is I think hugely important. But what can you tell us anything else about your company, or is it?
Yeah, so that's on the first technology, the CERTS technology, the protein-based system. So it is still in stealth, so I can't really talk a lot about it. But yeah, no, I think broadly speaking, we are these sorts of projects only have, we learn interesting things along the way, but they really only have an impact if they actually go and help someone. So I think as a lab, this whole subgroup in my lab is very motivated on thinking about, what we can do to have an impact. And again, it's with biotechnology, it's a complicated set of questions, picking out the areas that have an unmet need, but that also, frankly, have a market, so that someday there are enough patients that can be identified, and organizations like this can actually pull out patients to ultimately run clinical trials is really critical to ever making a therapeutic, and then having reasonable endpoints for those, and then thinking about, as a lab, we think a lot about where to focus our attention to try to make that impact. It's really difficult, but I'm totally aligned with that for sure.
You're touching on so many cool things. I think patient identification is a huge problem, right? And the good news and bad news on SYNGAP. The good news on SYNGAP is the data, the genetic models, and like Dennis Law's written on this, and I can send you the papers if you haven't seen them. There's a lot of data to suggest there's a ton of SYNGAPians. Good news. Yeah. Bad news, clinically, they're hard to find, right? They don't start seizing at birth. The phenotype progression is slow. So that's like the bummer. Like it's going to be work to find these patients. The good news is I have a list of like 2-year-olds, 1 or 2-year-olds, and there's like 10 of them now, because what's happening is kids are getting sequenced at age 1 as soon as there's delays or like people are catching seizures. And that was unheard of 5 years ago, right? So the rate at which we're offering genetic testing, thankfully, is going up. Now we're losing a ton of kids to missenses, which get bust, but that's a separate conversation. And then the question is, how do we figure out if therapies like yours, how do we figure out which missense mutants therapies like yours can help, right? Like that assay of understanding, is a missense dominant negative? Is it a null? Is it something in between? That is a huge rabbit hole that we don't have to go down right now. Okay, so JR has put 2 questions in the chat. I want to ask you about delivery, but I'll go to JR's questions first. Do you want to read them? Because
yeah, sure. Let's see. Comment on human variation. If the 3' UTR is highly conserved, then we'll get binding alternatives, both alleles for whatever RNA didn't get MD. If the 3' UTR has more variation, each patient needs to be sequenced, and perhaps the allele can be separated. Yeah, that's a great question. The heterogeneity of the 3' UTRs, I don't know the answer to that. Yeah, I think that we haven't seen indication. I don't think we would get lucky enough to find mutants in the 3' UTRs that correlate with the disease mutation that allowed us to get specificity. Usually one nucleotide or so, which is what the most you would ever hope for, probably isn't enough to differentiate. So we probably can't use it in a positive way. In the negative sense, though, if a patient had a mutation that precluded binding of our system, that would be a problem. So I guess, you know, you would diagnose the patient in terms of what their mutation is driving the disease, and at the same time sequence their 3' UTR and make sure the system actually can bind to it. So I think that would have to be part of the pipeline for how you would identify people. Let's see. Next question is how much NMD for each PTV is a total variable based on intron exon boundary distance and other issues. Sorry, what? I don't know. Can you write what is NMD?
Nonsense mediated decay.
Oh. Well, so we're not targeting nonsense mediated decay. We're targeting direct promotion of translation. So this is not related to the oligos that try to promote translation or try to promote gene expression through nonsense mediated decay. We're not doing any of that. This is completely orthogonal to all those, like, you know, whatever, Tango and all those sorts of things. So the RNA is there, and we're just telling the cell, make some more protein from this RNA. So totally different. So yeah, it has nothing to do with introns, exons. This is after splicing. So do you think both alleles will be there? Yes, both alleles are there. So in principle, if we target the 3' UTR, it will target the disease and the healthy, and they will both go up. That is definitely a potential challenge that we would face. But that's one of the reasons we went for SYNGAP1, because it looks like that would have clinical efficacy.
So let me just rephrase that for the mortals who are trying to follow along. DNA makes RNA makes protein. The people with protein truncating variants have a variant that would cause a premature stop codon that at the protein level would cause that protein to be spit out by nonsense mediated decay. But at the RNA level, both the good copy and the bad copy, the wild type and the mutant allele, will both have RNA, and your technology will attach to the 3' end, the back of the good and the bad copy, and make more of both.
Yes.
Is that right?
That's right.
Okay.
Yeah.
Very
so
cool.
again, thinking toward the future, like if you had a way to, and people are working on this for gene duplication based disorders, if you had a way to knock down the mutant disease, you know, dominant negative copy, that's a viable strategy there. And then potentially that would be what would be needed. If it turns out that that's the main issue for SYNGAP, then you would combine it, right? Knock down the disease causing variant or the disease causing mRNA, promote production from the healthy. But yeah, I think we have to see. Like I said, there's a lot of evidence that appears that the dominant negative aspect of it is for many mutations is probably less critical. And the other note about how to eventually commercialize or translate things like this, I mean, what I also personally find interesting about SYNGAP1 is there's GWAS data that suggests that there might be a much broader population of, say, autism patients that are not direct mutants in SYNGAP1, but that are SYNGAP1 deficient for other reasons that would also benefit from a SYNGAP1 restoration therapeutic. So in that case, they don't have a mutant allele per se, but they may be SYNGAP deficient and have autism for a very similar reason. So I think thinking about a pipeline for how would we ever make a therapeutic for autism, I've kind of bought into this idea that one way to do that is you pick a semi-rare disease like SYNGAP, you get a therapy for it, and then you just see, does this help with a broader population that you can identify through other sorts of metrics. So I think—
you give an example of why somebody would be SYNGAP deficient if they weren't a mutant.
Well, we don't know. I mean, so human gene expression is such a it's environmental factors, all the other mutations within that patient that changes gene expression. So it's a little unclear. But if you look at GWAS data, kind of there are things that point to the SYNGAP1 allele, the SYNGAP1 gene for various reasons. So
very cool, very cool. I might follow up with you on that and ask you to point me to something that puts that in writing. I mean, there's a whole other group of people who are like tau and SYNGAP definitely interact. And I could set up a debate right now. I can tell you exactly who will argue that tau is pushing down SYNGAP and people who would argue that SYNGAP is pushing down tau. And these two people not agree on anything. But I like your idea as it applies to the autism community, because certainly SYNGAP, unlike, the channelopathies, is a synaptopathy, is a really complicated gene, which actually gets me to my next question, right? One of the complications of SYNGAP, aside from the fact it acts in the synapse, and it has so many different functions, right? Is it structural? Is it functional? Whatever. There's at least 11 isoforms. And there are some therapeutic strategies that say, this is the isoform that matters. Let's just make more of it. Or I won't go down this rabbit hole. And some people are like, well, don't mess with the isoforms until you understand what they all do, because you could be creating other problems, right? Your therapeutic approach leaves isoform selection to the body. Is that right?
That's right. Yeah, exactly. As long as we pick the right site to bind to in the 3' UTR, then yes, exactly. It'll be reflected by whatever the content of that cell is that you deliver to.
So given that the 3', depending on where so in a sense, because the 3' UTR is part of what chooses which isoform.
Yeah, so we could in principle design them to target something that's isoform specific, but therapeutically, probably the best strategy would be pick something that is broad, that's present in as many contexts as possible, so that then you let the cell decide which ones are expressed. And you assume, in this case, I should say, that like the healthy allele is being expressed at the levels it should have been expressed at based off of the cell context. I mean, that is kind of a key assumption that we're making with this sort of strategy.
I have to think more about that, because I remember your slide, you had the G1, the G2, the G3, and the G4. And I'm just thinking about, depending on which G you connect to, what is the downstream implication of, will certain transcripts, I don't know if that's the right word, or certain RNA, certain RNAs will then be upregulated, but what would that mean for ones that were to the left or to the right of that in terms of isoform selection?
It's just a matter of whether those guide sites are binding, whether that sequence is present in the mRNA that you're targeting. That's the only question. And that's, and I guess one advantage of the system is we have a lot of flexibility, so we can, we can kind of target. We can't target any site in 3' UTR, but if we have, it's huge, right? There's hundreds and hundreds, thousands of bases. So we have a lot of different options that we can kind of go after within the 3' UTR. It's not like one site that we need to target.
Cool. All right. Then JR has another great question. I want to ask you one more.
Yeah, so the question is delivery, which is the correct question to answer. And just big step back from me thinking about biotechnology, right? Any therapeutic involves three things. It involves the technology, which is what I get really excited about. How are we going to do cool stuff to a cell that other people hadn't done yet? The other one is the patients. Do patients actually need this? And what are the needs that they can kind of, what's their pathology and what are they willing to accept from a therapy? And then third is the delivery strategy. How do you actually get that cool technology into those patients? And I think those three things are all equally important when thinking about a new therapeutic technology. So in this case, like I said, my lab is really focused on the technology. This is what we care about. We think about those other two, but the question is what is viable, right? If we're going to go after, one of these kids that doesn't have another option, then the question is, what are they willing to try, right? Is an intrathecal injection a viable option? Long-lasting oligo, do we really need to get to, is a gene therapy type approach with AAV viable? Then maybe our CERT platform would be an alternative mechanism for this. Or do we really need an orally bioavailable compound and really a small molecule, in which case these are really just stepping stones toward an ultimate kind of small molecule drug that one would take via pill. And if that's the case, it's going to take a lot longer to get to that. So my long answer is, I think from the academic lab, we get the technologies, and then the idea is eventually we hopefully start companies that think through these issues and what's the quickest way to get help to patients. Specifically in this case, though, what we're doing in the lab is we're testing things via AAV when that makes sense. We're testing things via RNA delivery when that makes sense. The brain is really tough, as you probably know. We can get oligos in the ASOs for splice switching things like the nonsense mediated decay. There's some evidence that suggests those can work in the brain. Lipid nanoparticles don't work well in the brain. Unfortunately, they work great in neurons in culture ex vivo. They work really poorly in the brain, but there are other delivery strategies and oligos making their way into that. And then I would just say, maybe from a more optimistic view, there are so many interesting oligo technologies being developed. I talked about some of them. There's just many out there. So I think in the next couple of years, the hope, at least, that we're counting on is that more strategies will emerge, more kind of positive clinical data will come out, and there'll be other options for how to actually get things delivered effectively. But, long term, we're not counting on that. Like I said, we're investing some of our resources in thinking about other types of modalities to go after, to move away from oligos. We have protein-based systems and viral delivery. That's a really quick way to get into a patient that we could do very rapidly, but has its own limitations. So there's no right answer. It's a matter of control and technology. And then, like I said, what patients actually want and what, how fast you want to move into the clinic, I think.
Yeah, it's a rich answer to a loaded question. I mean, so let me take the willing part head on. Our kids, kids with SYNGAP turn to adults with SYNGAP. And I can share this data with you if you're curious. There's a poster out of CHOP that beautifully illustrates that on the behavioral and the autistic features of SYNGAPians as compared to Rett, as compared to Angelman, as compared to epilepsy cohorts, we are like way above the mean. Right. So our patients our patients suffer mightily. They're very challenging, wonderful human beings, but they're very challenging to manage. And it takes a huge toll on the humans, the families, right? All rare disease families struggle, but the behavior part makes it really hard for our families to get help, because people kick our kids out of programs all the time because they just don't understand what's going on with our kids. So I think the willingness to take on therapies, be it intrathecal or whatever. Obviously I can't speak for everybody, but one of the reasons I do what I do, and I'm constantly talking and I'm constantly telling people about people like you, is because I want families to understand that they will have a chance to do an intrathecal injection at some point in the future, and they should take it very seriously, right? They have to understand, but that's part of why we're having this conversation right now. I want people to be thinking about this. So I would go as far as to say, I believe we can find families who will be willing to take these therapies for their kids, because as Gavin Rimbaugh said in our last scientific meeting, neurons want their SYNGAP back. And we believe that the brain is plastic enough, and that the way the synapse works is if we restored SYNGAP, we could, kids aren't going to become neurotypical, but they could seize less, they could sleep more, they could be less anxious, they could be easier to manage. So I think there's tremendous willingness and incredible need here. And I would encourage any family member on the call to chime in and tell me if you have another perspective or if you agree. On the delivery thing,
just a quick note of that. I mean, honestly, that's really helpful to hear. I mean, from our perspective as like basic technologists, it's really hard. Sometimes it feels overwhelming to do this sort of work because you do have to think about so many things at once, and it seems intractable. So we can develop technologies, but, having a sense that, okay, we can actually test these ideas, and there's a short-term goal, right? And maybe a medium-term goal. It really is, I think, very helpful to make the scientists feel like they're empowered to actually try something, because these seem, like I said, they just seem intractable, but I don't think they are. I think where the world is at right now, there are solutions that can exist.
They're not, and we're willing. I mean, I'm just looking through the participant list. Like, we have parents of an 18-year-old, a 4-year-old, a 2-year-old, another 2-year-old, and a 17. I mean, all ages, all of them. Everybody's eager for help and wants to understand this. I want to talk like delivery. This is something that's really hard for me as somebody who sits and gets proposals from everyone and their mother, right? All these cool technologies, right? From Tango to what you're doing to other cool stuff. And then you have people who are talking about different delivery mechanisms, and they're like, ASO, intrathecal, keep it simple. And then other people are like, this AAV is good. This AAV's gone through AAV9. Everybody's used it. Let's just use it. AAV9 sucks. Doesn't get to enough of the brain. AAV, pick a number. This is better. We're doing it in my lab. 2 years. This will be better. And I'm just like, oh my, you're all making my head hurt. And at the end of the day, I think it's going to be both, right? Like, I think you guys figure out how to make an elegant, efficient, minimal off-target effect solution. And then there's a lot of good people working on a lot of delivery systems. And then the question is, who's going to, we just have to hope, pray / introduce you to each other, that you guys meet in the middle. And, people like Al George and others know a lot of these players that can say, hey, you should talk to, for instance, we know that at Penn, there's this wonderful grant where they're doing a lot of SYNGAP work, and Bev Davidson is working on some AAVs. So like, if you're thinking about potential partners, the Penn team is thinking about AAVs and SYNGAP delivery. And I think delivery to the brain is a whole ball of wax that hopefully people are thinking of. But I think what's loaded in JR's question, this is cool, is she mentions gut, pituitary, and other systems. So what if, which obviously JR and others believe, SYNGAP actually affects outside of the brain? And it's just called SYNGAP because, Rick found it, and Rick's a neuroscientist, so everything's the brain and nothing else matters, right? God bless Rick. And so, but what if we need SYNGAP in other parts of the body? How would that work? How would we deliver this to other parts of the body?
Yeah, it's tough. It's really tough. If it was the liver, it'd be easy. The liver's the one place we can deliver things to super efficiently with lipid nanoparticles.
Because everything goes to the liver, right?
Everything goes to the liver. But yeah, other places, it's really tough. Honestly, it's really tough. I think, and probably my guess is mouse models, I don't know, but probably wouldn't even be, sensitive enough to see some of those effects. Honestly, and to your answer, I do think that all these delivery strategies each have validity. And I think, they'll each find their right application, maybe the right patient subset. So there's not going to be, at least not in the short term, one answer. I think in 100 years, I'm a chemist, in 100 years, we will take everything we learned from all this and everything will turn back into a nice small molecule drug pill that we can take and just have the defined activity. It's going to take another century to get to that. So it's really just these intermediates, right? Where we need to do viruses or RNAs in this case, or these things where we're not good enough at designing molecules that have more complex function yet. But I don't know. I think a lot about this. It's challenging. You could do AAV in other tissues, but systemic whole body AAV delivery, I think, is not, that seems like a pretty big ask, at least for a first generation of a clinical trial. You wouldn't want to do that, I don't think.
Totally. I think in the short run, we deliver to the brain. I think and I would encourage, if not you, definitely, I think you've said his name was Riley, whoever's in your lab, if they can't come in person, and if they need help coming in person, call me. But they should definitely follow the live stream. We have an incredible day of science on November 30th, science and clinical readout. And whoever in your lab is thinking about SYNGAP in the morning, that's going to be a great meeting.
Awesome. That's really great.
With that, I'm grateful for your time. JR generally says it best. That was a beautiful presentation, very lucid, really cool tech. And
Thanks.
JR also echoed my point on these kids need help. So we're glad you've chosen SYNGAP, and we look forward to staying in touch. Last call for the audience. If you type now or forever hold your peace. Okay.
Yeah, no, thank you all. And I'll pass on. It's really helpful. Again, the grad students are actually in the lab doing these sorts of things. I think, like I said, everyone on this team, I think, is really motivated about trying to use they're all in their 20s, using the best years of their life, spending long hours in the lab, trying to do something that has some off chance of benefiting the world. So it's really helpful to know that there are people on the other side of what we're thinking about. So I'll definitely pass that on to them. And if anyone wants to reach out, we'd be more than happy to make more connections. It's really important for what we do.
Great. Thank you so much. This was
okay.
awesome.
Thanks so much for the invite again. It was really, really a pleasure.
Same here.
All right.